And the first type of innate defense, as we already talked about, are those physical barriers. Probably the most easy to understand would be the skin. You also have the mucous membranes, and those are going to be lining the respiratory tract and the digestive tract, for example. So those are also places where you can have pathogens possibly invade the body, so you have to have barriers in those locations. And you can also think of other physical barriers like the blood-brain barrier, for example. All of these are essentially denying access to the body of the pathogens. The secretions on top of these barriers may have bacterial-side or antibacterial agents in them, for example, secretions on the surface of the skin. And all of that is important to prevent pathogens from ever even entering the body. So this is really your first line of defense. If the pathogen does get into the body, there are some internal innate defenses. And so one of those is our second innate defense that we're going to talk about, which are phagocytes. And there are different types of leukocytes that can act as phagocytes, which we've already learned about. These include your macrophages, like your neutrophil, your eosinophil has phagocytic ability, and of course you have your monocyte, which when activated becomes the big eater. Macro means big, and phage means to eat, so big eater, macrophage. So a phagocyte is a cell that's going to ingest these pathogens and essentially destroy them. Not only is it involved in innate defenses, where ingestion and destruction of the pathogens is essential for clearing them out of the body, but it can also activate the adaptive defenses by letting the lymphocytes, the B cells and the T cells, know that the body has been infected, and we'll talk about that in later lectures. This is essentially how phagocytosis works. You get endocytosis of the pathogen here, because the phagocyte is going to adhere to the pathogen and then be taken in into a phagosome or a phagocytic vesicle. And that vesicle is then going to fuse with a lysosome, which you will probably remember is an organelle within the cell that contains hydrolase enzymes. Those enzymes are going to break down the pathogen into small pieces. So toxic compounds and lysosomal enzymes destroy the pathogen. Sometimes exocytosis of the vesicle removes indigestible and residual material. But note that the macrophage can take pieces of that pathogen and put it out on their membrane like a flag to let the body know what we've been infected with and activate the adaptive immune system. So we'll be talking about that soon enough. A third type of innate defense is called the immunological surveillance system, and this involves the natural killer, also known as NK cell. This is actually a type of lymphocyte, and it's the only type of lymphocyte that's involved in innate defense and not adaptive defense. So the other types of lymphocytes are the B cells and the T cells, which we will talk about later. So this involves the natural killer cell, the NK cell. And what the NK cell does is it's going to recognize an abnormal cell. This cell has these abnormal surface proteins on it, suggesting that it's either foreign or cancerous. It just does not belong in the body. And because of that, what it does is it aligns its Golgi apparatus to the cell where it's attached itself. And it essentially secretes molecules into the cell, and these molecules are called perforin because they perforate the cell's plasma membrane that it's attached to. So here are the vesicles coming from the Golgi apparatus containing these perforin protein molecules. Exocytosis occurs and the perforin molecules go to the abnormal cell membrane, and they basically insert into the membrane and form holes and it's like punching holes into a cell, and you all know that the cell membrane is very important in regulating fluid balance in the cell, and if you just punch these holes in there, the cell, it's going to cause the cell to lyse or to burst. So it's killing the other cell. That's why it's called the natural killer cell. So that's another type of innate defense. They don't go after specific types of pathogens. They just notice that a cell is abnormal in general. The next type of innate defense is the interferon. Interferons are actually really quite cool molecules. Interferons are released by either activated lymphocytes, macrophages, or infected cells. Lymphocytes, macrophages, or an infected cell, which is what I'm going to talk about as an example here in just a second. So all of these are going to secrete these molecules called interferons. And these interferons are going to go and signal adjacent cells or nearby cells. And when they signal the cells, they bind to a receptor and they tell the cells to produce antiviral proteins. So interferons are usually defending the body from viruses. And this diagram right here is a really nice example. So here's a virus, and a virus is not a cell, it's just basically packaged up nucleic acid. So in the package is the nucleic acid, and what the virus does is it inserts its nucleic acids into the cell, and that causes the cell to be infected. The virus is going to basically take over the cell and get it to start manufacturing viruses for the cell. So it becomes a viral factory. But the virus particle also, nucleic acid, also is going to cause genes within the cell from the normal genome that create interferon. And the interferon is going to be packaged into vesicles and secreted and act as a paracrine factor, which means it acts only on local cells. So it's going to go over and bind to adjacent or nearby cells, to receptors, and when it binds to those receptors, it activates genes in that cell which produce antiviral mRNA and then antiviral proteins which block viral reproduction in the nearby cells, meaning that the nearby cells are protected from invasion of that virus. So the infected cell itself is, although it's able to produce those interferons, it's going to die because it's been infected by the virus, but it's able to keep alive its neighbors, which is good. Fifth type of innate defense is the complement system. The complement system can also be activated through adaptive defenses, so it can be activated in either type of immunity. So through adaptive defenses, it can be activated by antibodies, which are produced by B cell derivatives. It can also be activated directly by lectins, which are molecules that bind to specific sugars on the microorganism's surface, or just spontaneously because that microorganism lacks a type of surface and it allows the process to proceed. So all of these things are going to cause the complement system to be activated. Complement system is just basically a group of plasma proteins that come together to form a complement, and they can have different effects. One thing that they do is called opsonization, and this is when, if you have a bacteria, the complement proteins can come over and they form a coat around the protein, sorry, not around the protein, around the pathogen or the bacteria, making it easier for a macrophage to come along and engulf it. So that's called opsonization. They can also enhance inflammation by causing the release of histamine, which increases blood vessel permeability. And we'll talk about inflammation a little later. Another really cool thing that they can do is they can form membrane attack complexes, or MAC for short. And MACs do this. So they insert into the target cell membrane, creating pores that lyse the target cell. So it's very similar to the activity of perforins, actually. A fever is when your body temperature rises above normal body temperature, which is 37 degrees, in response to molecules called pyrogens. So what is a pyrogen? So a pathogen itself might be a pyrogen, and a pyrogen is anything that causes the body to have a fever, meaning that it homeostatically defends a higher body temperature. The set point for body temperature actually increases. So you're defending a higher body temperature during a fever. The reason for that is that these pyrogens may activate white blood cells, leukocytes, which will secrete molecules, for example, interleukin 6 is a common one, which act on the hypothalamus, which sets the body temperature. And normally body temperature is going to be around in Fahrenheit 98.6, but because of the change in the set point, you're going to get a higher body temperature of 103. Well, there's many theories about why you actually would want to do this. For example, this may mobilize your defenses. Your white blood cells might act more quickly because of an increase in metabolism. This might accelerate repair of tissue, and it might actually inhibit the activity of some pathogens, virus or bacteria. All right, last one, inflammation. So this is a very detailed diagram that you can read through. I'm not going to read through everything, but essentially if you have tissue injury or invasion of a pathogen, your mast cells are going to secrete chemicals, for example histamine and prostaglandins, which are going to do things like cause your arterioles to dilate. That leads to increased blood flow to the area, which causes it to be warm and red, things you associate with inflammation. You're going to get increased capillary permeability. This leads to an edema and swelling. All of these things can lead to pain. And then you're also going to get a release of leukocytosis inducing factors, which increase white blood cells. And white blood cells are going to migrate to the injury, and that's going to help clean up the dead tissue, clear the debris, and all of this will promote healing of the region.